This is DNA, or deoxyribonucleic acid. It's in the shape of a double helix. So the string that's twisted is our DNA, and the purple segment is a gene which codes for a peptide. First, the transcription factors, which you can see in the black, come and bind to the TATA box, or the start part of the... I can't do it. Wait, I forgot what
Take one.
Called. And here are the nitrogenous bases. Orange, cytosine.
1.
Red, adenine. Blue crayon, thymine.
1.
Blue candle, uracil. Green, guanine. In DNA, the nitrogenous bases always pair with the same ones. So A and T pair together, and C and G pair together. And the strands are anti-parallel, so the 5' end is with the 3' end on the other side. In mRNA, thymine is replaced with uracil, so the blue crayon is replaced with the blue candle. Initiation is after the RNA polymerase binds to the DNA. It starts the RNA synthesis at the start point of the DNA template strand, so at the 5' end. Here, during elongation, the RNA adds on to itself in the 5' to 3' end as the RNA polymerase continues along the DNA and unwinds it. The DNA behind the RNA polymerase rewinds back into a double helix after the RNA polymerase passes it. Once all of the RNA is long enough and elongation has ended, termination will occur when the RNA polymerase releases from the DNA, and the mRNA that has been formed also gets to go away. Pre-mRNA must go through some processing before it can be cut into just mRNA. So, in pre-mRNA processing, enzymes add a 5' cap and a poly A tail to the pre-mRNA. The 5' cap is modified guanine sequences, and the poly A tail is adenine sequences. The 5' UTR is the untranslated region of the 5' end, and the 3' UTR is the untranslated region of the 3' end. The middle part between the start codon and the stop codon is the protein coding segment.
1.
Once the pre-mRNA is transferred into the cytoplasm, it must be altered into being mRNA. So pre-mRNA has exons and introns. Introns are intervening sequences, and they must be cut out because they do not code for any part of the protein. So snRNA and a protein and other proteins come together to form a spliceosome, which is represented by the scissors. And the spliceosome attaches on to the pre-mRNA and cuts out the intron pieces. So the intron pieces are no longer needed, and the exons and the 5' and 3' caps come together to form a finalized mRNA.
1.
Through translation, each exon comes to represent one part of a domain or one part of a polypeptide that ends up making a whole protein. A ribosome is made up of a combination of RNA and proteins, which are molecules called ribosomal RNAs, or rRNAs. A ribosome also has a large subunit and a small subunit. The large subunit is where the tRNA binding sites are, so the A, P, and E sites, whereas the small subunit has the mRNA binding site. A ribosome is where the process of translation occurs. In the ribosome, there are 3 binding sites, the A, P, and E sites. The A site holds the tRNA, bringing the next amino acid to the polypeptide chain. The P site holds the tRNA carrying the growing polypeptide chain, where the E site is the exit site. So the tRNA that has already dropped off its amino acid can leave the ribosome from there.
1.
Amino acids are all in the cytoplasm of each cell. There are 20 different amino acids, like the start codon amino acid methionine, or short for MET. Amino acids are the monomers of proteins. They all have unique R groups that make them fold and interact different ways, which is what creates the complex structure of proteins.
1.
tRNA, or the tape, is transfer RNA. Transfer RNA job is to bring amino acids like methionine from the cytoplasm into the ribosome so they can all be connected to form a polypeptide. tRNA on one end has the amino acid that is coded for on the other end by the anticodon. The anticodon matches up with the correct base pairings provided by the mRNA in the ribosome. The start codon of any protein is AUG, as we can see in the A binding site. So the anticodon is UAC. In elongation, the tRNA that has already dropped off its amino acid leaves the ribosome through the E site. In the A site, a waiting tRNA with an amino acid sits. In the P site, the anticodon matched with the codon in the P site brings another amino acid. In the site, a peptide bond forms between the amino acids, making this chain of amino acids a polypeptide. Eventually, a stop codon is reached in the mRNA. Stop codons include UAG, as seen here, or UAA or UGA. They do not code for amino acids. Instead, they code for a protein called a release factor, which is this marker, and the release factor comes and binds directly to the stop codon in the A site. In the P site, instead of adding an amino acid to the chain, it adds water, creating hydrolysis, which is what allows the amino acid chain or polypeptide to then be released from the ribosome, and translation and termination is therefore complete.